All right, today we're going to dive into a real medical detective story, one that takes place at the genetic level. It's all about a truly cruel, debilitating disease, but more than that, it's about the brilliant scientific puzzle that stood in the way of really understanding it.
This isn't just a list of symptoms on a page. It's like a slow-motion theft of a person's mobility and their senses. This disease, inherited peripheral neuropathy, or IPN for short, it just gradually weakens the muscles and damages the nerves in your limbs. It makes the simplest things we all take for granted, incredibly difficult. And the crazy part? It all starts with one tiny error, a typo coded deep inside our DNA.
And get this isn't some super rare, obscure condition. IPN affects about 1 in every 2,500 people. That's thousands of individuals, thousands of families whose entire lives get turned upside down by this disease. And that's exactly why the search for answers isn't just some academic thing. It's a deeply, deeply urgent mission.
And right here, this is the central mystery that had scientists totally stumped. They knew that mutations in over 100 different genes could cause IPN. But for a few of these really key genes, they saw something that just didn't make sense. Some mutations were absolutely devastating, while other mutations that looked pretty similar were, well, they were harmless. Why? What was it that made one tiny genetic typo a total catastrophe and another one nothing? That is the puzzle we're going to solve together today.
So to crack this case wide open, we've got to zoom way, way in. We need to take a quick tour of the microscopic factory that's running inside every single one of our cells. Because this is where the genetic constructions get read and carried out, and it's where our mystery really begins. The easiest way to think about it is like this. Your DNA, that's the master blueprint. It's locked away in a safe place. Now, when the cell needs to build something, it doesn't take out the original blueprint. No way. It makes a temporary copy, and that's called messenger RNA, or mRNA. That copy gets taken out to the factory floor, a little machine called a ribosome, which reads the instructions and builds the final product, a protein. And it's these proteins, they're the little workers that do pretty much everything in your body. Now, the most important workers in our factory story are a group of enzymes with a super long name, aminoacyl-tRNA synthetases. Yeah, it's a mouthful, so let's just call them ARS for short. Their job is absolutely critical. They're kind of like chargers, making sure the right building blocks, the amino acids, get attached to their delivery molecules. If an ARS enzyme messes up, the whole protein assembly line can just grind to a halt, or even worse, start making faulty products. And it's mutations in the genes for these specific enzymes that are linked to this disease. So this brings us back to the big question, right? How can one little mutation in an ARS gene be so destructive while another is totally fine? Well, scientists came up with this really elegant and honestly kind of sinister idea. The title here says it all. What if, in this genetic equation, 2 was somehow less than 1? All right, let's just dust off our high school biology for a second. For just about every gene, you've got 2 copies, one from mom, one from dad. So if you've got one healthy gene and one faulty one, you'd probably think the healthy one could pick up the slack, right? Well, it turns out it completely depends on how that other gene is faulty.
Okay, this is where it gets really fascinating. On the one hand, you have what's called a null mutation. That just means the faulty gene produces nothing. It's a blank. It's like one worker calls in sick. The other, healthy gene, keeps on working, maybe at 50% capacity, which a lot of the time is good enough. But a dominant negative mutation? Oh, that's something else entirely. It's not like the worker just called in sick. It's like the worker shows up to the factory and actively starts breaking the good worker's tools. It's a saboteur.
So how does this sabotage actually work? Well, these ARS proteins, they have to pair up to function correctly. They work in teams of 2. But when a faulty, mutated protein pairs up with a healthy one, it creates what the researchers perfectly called a broken duo. The faulty partner basically poisons the pair, making them both totally useless.
And wow, this chart just lays it all out perfectly. Look at the left. With 2 healthy genes, you get 100% enzyme activity, just like you'd expect. In the middle, with one healthy gene and one that's just absent, a null mutation, activity drops to 50%, still often enough to be okay. But now look at the right. With one healthy gene and one of these saboteur mutations, activity plummets to less than 50%. The saboteur isn't just missing in action, it's actively dragging the healthy one down with it.
Okay, so that's a brilliant theory, right? It makes perfect sense. But in science, a theory is nothing without proof. So, to test this whole sabotager idea, researchers turn to one of the simplest and frankly most powerful tools in genetics.
Yep, you heard that right. We're talking about the same simple yeast you'd use to make bread or beer. Why yeast?
Well, yeast cells are simple. They grow incredibly fast, and their genetics are pretty easy to play around with. That makes them the perfect little living laboratory to test what's going on with these human genes. And the experiment they designed was it was so elegant. First, they engineered the yeast so they could switch off its own ARS gene. Then, they gave it the human ARS genes and made it rely on those to survive. Now look at that top row. When they gave the yeast one healthy human gene and a null one, the yeast grew just fine. That one healthy gene was enough to do the job. But, and this is the moment, look at the bottom row. When they gave the yeast one healthy gene and one of those neuropathy-causing saboteur mutations, the yeast just failed to grow.
I mean, that was it. That was the smoking gun. The experiment proved it beyond any doubt. The neuropathy mutation wasn't just broken. It was a saboteur. It was actively interfering with the function of a healthy gene, exactly like the theory predicted. The mystery was finally solved. And look, this discovery is so much more than just a really neat piece of science. By understanding exactly how the system breaks down, it shines a bright light on a clear path forward, a path for developing treatments and bringing real hope to thousands of patients. This also perfectly explains why the symptoms show up in the arms and legs. You see, the long nerves that run all the way down our limbs are like these incredibly long supply chains. They have to keep making proteins at their very ends, which are really far from the cell's main body. This makes them super sensitive to any slowdown in that protein factory. A small drop in ARS activity that might not even be noticeable elsewhere in the body can be totally catastrophic for these long, hard-working nerve cells. So this is where the real hope comes in. Now that we know the enemy is that faulty protein, the saboteur, we can actually design therapies to take it out. Can you imagine a drug that could find and just block the faulty mRNA instructions before the saboteur protein is even made? Or maybe a treatment that disables only the faulty protein, letting the healthy one finally do its job. And what's really cool is that those clever yeast strains from the experiment, they can now be used as a super fast screening tool to test thousands of potential drugs to find one that actually works.
And this whole story leaves us with one last huge question to chew on. This whole saboteur effect, it solved the puzzle for this one disease. But you have to wonder, how many other unexplained genetic conditions out there might be caused by the exact same thing, a single faulty protein actively messing with its healthy partner? Answering that could unlock treatments for diseases we don't even fully understand yet.